There's an amazing field that's evolved rapidly over the last few years. I'm talking about the human microbiome, the population of commensal bacteria that live on us and inside us.
Yes, I'm sure that as a gastroenterologist, GI microbiome is very important to you. It has been evident in the past few years that it affects human health and disease.
Well, not just GI conditions, which are closer to me, like inflammatory bowel disease or non-alcoholic fatty liver disease, but also, surprisingly, in organs that are distant from the gut. For example, a link between the human microbiome and Parkinson's disease was recently reported in Cell.
Clearly, the development of understanding the microbiome has relied on several technological breakthroughs that happened in the past decade or so. But in order to understand them, one needs to first understand the scientific context. The big picture here is a new sequencing technology, what we call next-generation sequencing, or NGS for short. And it's much bigger than the microbiome. It's not only for that, it's a whole new sequencing technology.
How so?
Let me explain with an analogy from a different revolution, the invention of the transistor that replaced the old vacuum tube triode. It was a pivotal change that enabled the transformation to submicron dimensions. This revolution is what enabled modern electronics as we know it today. Similarly, the ability to sequence humans, which was a huge undertaking until the first Human Genome Project was completed in the year 2000, now became, with the modern technology, quite routine. And more applications are added every day now.
So the same technology is used to sequence both bacterial and human genetic material?
Yes.
You'd think that it would be more simple to sequence a bacterium which has a genome of only a few million nucleotides than a human genome which has 3 billion.
Yes, but the issue here is not to sequence a single bacterium, but actually to sequence simultaneously a population of bacteria which can be comprised of 1,000 species that live, for example, in the gut.
You know, many of these bacteria can't be grown in the lab, so in fact what is done here is sequencing of a mixture of genomes of unknown organisms, which is a real challenge.
Yes, the next generation sequencing technique is actually very complex and comprised of both molecular and computational technologies. I think it is important to understand both of them in order to really get understanding of the next generation sequencing revolution.
Ron Unger.
For the last time in this course.
Ron can give a fantastic explanation about the NGS technique, the computational parts, and some advanced applications.
He will. And I will see you next time in another Spotlight session.